I will talk about a publication that deals with a small proof of concept experiment on artificial intelligence and new genomic techniques that was recently published in Frontiers in Plant Sciences. It shows that AI can use the NGT1 criteria of the EU proposal on plants obtained by certain new genomic techniques as a design room to escape mandatory risk assessment for genetically engineered plants. And it shows the implications of the convergence of AI and NGT for the proposed future regulation of NGT plants. I will start with the requirements enabling the convergence of AI and NGT, which was made possible primarily by two technological developments. First, in recent decades, large amounts of biological data have been digitalized and made available in databases like data on whole genomes, RNA, proteins, metabolites, microbiomes, and pangenomes. And AI can browse vast amounts of these big data in seconds for the design of new gene variants and gene combinations. On the other hand, a wide range of new genomic tools and methods made it possible to genetically engineer almost any gene in any life form. And this has led to the development of a variety of special AI algorithms and platforms that can be used, for example, to locate specific genes, identify regulatory elements, discover specific gene variants, and predict the outcome of genetic engineering interventions or the combination of multiple genetic changes. In addition to the most frequently used CRISPR-Cas9 nuclease, more advanced recombinant enzymatic mutagens now enable more specific deletions, like, for example, the already mentioned Cas12 nuclease. So-called base editors can exchange single key nucleotides, and with prime editors, it is possible to introduce small artificial DNA sequences. So as already explained in detail by Franziska, NGTs can overcome many constraints posed by evolution, which has led to a change in the approach of genetic engineering that has taken place in recent years. Instead of directly engineering the genes of interest, their expression is now more and more influenced indirectly by targeting small but powerful regulatory units, which are called cis-regulatory elements. And in this sort of fine-tuning approach, only very small changes in DNA are involved, often within close proximity to each other, which would hardly be achievable using conventional breeding. But the effects may nevertheless exceed the range of known plant characteristics, resulting in traits previously unknown in the breeder's gene pool. Cis-regulatory elements are regions of non-coding DNA, including promoters, enhancers, silencers, or insulators, which regulate the transcription of neighboring genes. And in many NGT applications, changes are now being introduced, for example, within the promoters to achieve scalable effects in gene expression by reducing or enhancing the transcription, as shown here in B and C. Cis-regulatory elements also include upstream open reading frames, short UOFs, which are located upstream of the main open reading frame and are highly conserved among plant species. And they act as translation initiation inhibitors through ribosome stalling at the mRNA and thereby reduce the production of the corresponding plant proteins. So consequently, the knockout of upstream open reading frames will lead to higher levels of the respective protein, as shown here in D. So after these more general considerations, I now come to the proof of concept experiment mentioned in the paper published in October. In the publication, we have integrated this trend of targeting small but effective regulatory elements with only minor changes in the genome to, with the help of AI, design the genetic blueprint of an insecticidal NGT plant. And this insecticidal NGT plant, according to the criteria in Annex 1 of the EU proposal, would not require mandatory environmental risk assessment prior to release and marketing, even though it may obviously pose serious risks to biosafety. To this end, we used the publicly available ChatGPT-4 or deep research agent to design the genetic blueprint of an insecticidal NGT1 maize by permanently increasing the production of a plant's own defense protein with only a few changes in regulatory units and with the requirements or the design room for category 1 NGT plants of the EU legislative proposal as a guideline. And here you can see an overview of the technical steps, starting with an initial prompt of the basic idea. The chatbot provided several scenarios and offered various options for realization, with which it was possible to successfully design the genetic blueprint for either a promoter swap of the infestation-induced promoter of the defense protein with a constitutive active promoter to increase the production of the insecticidal substance, shown as option A, or the inactivation of repressive cis-regulatory elements of the defense protein, shown in B, to as well increase the production of the insecticidal substance. We then asked for detailed sequence information, for which the chatbot provided the corresponding specifications. First, for the design of the guide RNAs enabling the promoter swap of the defense protein, and the design of the exchange promoter sequences. And second, for the design of the guide RNAs enabling the inactivation of 3 repressive cis-regulatory elements of the defense protein, shown here for repressor number 2 in option B. And this resulted in a design for an in silico NGT-maize that contains a higher concentration of a toxic substance, where insects feeding on it may absorb large quantities of the toxic substance and die. However, what no longer needs to be investigated due to the absence of mandatory risk assessment for these NGT plants is: what are the effects of the changes in toxicity on non-target insects that, for example, feed on the pollen or on soil organisms and their interactions with the plants? Are there unintended effects in plant composition and metabolism that may affect food safety or its nutritional characteristics? What are the consequences for the food webs and ecosystems? And what about the risks of gene flow and the development of resistance in pest insects? On the next slide, I would like to show you an example that illustrates quite clearly that the development of the convergence of AI and NGT has already reached a stage where there are even now, so to say, all-in-one solutions for the automation of NGT design and data analysis, as shown here for CRISPR-GPT, which was recently developed by leading US universities and Google DeepMind. CRISPR-GPT is a multi-agent system that should assist users in selecting suitable CRISPR systems, experiment planning, choosing delivery methods, drafting protocols, and so on, enabling fully AI-guided gene editing experiment design and analysis. As shown in the overview, one can select different NGT scenarios and different modes of interaction to streamline experiment design and planning, and even an evaluation tool to assess the performance of the agent is also integrated. Therefore, this tool is a professional version of combining generative AI and NGT, which will enable results that we more or less conducted manually in our proof of concept experiment. And since CRISPR-GPT is just one among many such programs, it can be assumed that this will become more and more standard lab practice in the future. So while it is often discussed that AI may be abused by malicious actors, causing major biosecurity risks, like developing dangerous viruses, our example demonstrates risks to biosafety, meaning risks for the environment and biodiversity. And it shows that more attention has to be paid on the protection of biodiversity and food production. AI now allows to rapidly search large databases to identify and generate specific genetic changes, especially in regulatory units. And this expands the design room for the generation of previously unknown gene variations, particularly in combination with new types of recombinant enzymatic mutagens. Therefore, the amount and complexity of those fine-tuned NGT plants can be expected to increase, making them highly relevant for the future regulation of NGT plants. And taken together, the significance and use of AI in developing NGT plants and other organisms, especially in combination with recombinant enzymatic mutagens that can overcome constraints in plant breeding, is evident and already taking place, but has nevertheless not been taken into account in the EU proposal, making it already outdated before it has even been implemented. And since several companies are already using AI to develop NGT plants, it has to be assumed that their algorithms are much more effective than a publicly available version of ChatGPT. And with this, I'd like to thank you for your attention and look forward to the discussion.